In this set of slides, I want to give you a very brief introduction to carbohydrate nomenclature, carbohydrate structure, and a little bit of carbohydrate biology, realizing that we have not nearly enough time to cover this subject in the detail that it warrants. So, first of all, carbohydrates are hydrates of carbon. That is to say, for every carbon atom, we have this water molecule that's associated with it, effectively hydrates of carbon. That's an old definition, and we'll see that it falls apart sometimes, but it's a good place to start. In any case, carbohydrates are molecules which can have multiple chiral centers, at least in the case of 4 carbons or greater, and we see in this case we have 2 chiral centers. Likewise here we have 2 chiral centers. Some definitions. Molecules that have such asymmetric centers can exist as diastereoisomers. In this case, we have a potentially 4 diastereoisomers because we have 2 different chiral centers around which we can have 2 different organizations. However, if we only vary one of those chiral centers, as we have in this slide, we refer to these as epimers. So these 2 carbohydrates are epimers of each other, in which we only vary the organization around carbon 2. However, the overall designation of these sugars is the same, and that's because, in terms of carbohydrate nomenclature, the overall designation is given by the organization around the penultimate carbon. So first of all, these are Fischer projections. Remember that in Fischer projections, we have a simplification in which we flattened a molecule that is not planar. The vertical bonds should be considered as going into the plane of the screen, the horizontal bonds coming out of the plane of the screen. The penultimate carbon here, in this case carbon 3, that is the carbon right before the last carbon. These are in the D configuration, that is by definition, hydroxyl on the right side is our D configuration. These are tetroses, so we use a sort of a Greek numbering system. They have 1, 2, 3, 4 carbons, so these are tetroses. And we number beginning at the carbonyl end of the chain. So this is an aldehyde carbon here. In this case, the carbonyl carbon happens to be the beginning of the chain, so that's carbon 1. But for these, we see these 2 different sugars. These are 2 carbohydrates that we'll probably have heard of at least before, fructose and glucose. Both of these are 1, 2, 3, 4, 5, 6 hexoses. Both of these have penultimate carbons in the D configuration, but we see that the carbonyl carbon is either at the 1 position in the aldose. Of course, it's an aldose because it's an aldehyde carbon. Here, the carbonyl carbon is a ketone, so this is a ketose. This is a ketohexose, ketose with 6 carbons. This is an aldohexose, aldose with 6 carbons. We'll refer to these carbonyl carbons as the anomeric carbons. And what is that all about? Well, it has to do with cyclization of the carbohydrate. Here in this cartoon, we show the linearized form, and we show what happens at equilibrium when we're free in water. We see that what happens is that the molecule does not exist to any great extent in this linear form. Instead, the molecule tends to cyclize. That happens when, in this case, the hydroxyl on the penultimate carbon attacks the carbonyl carbon of the anomeric carbon. In this case, again, that's going to be carbon 1, although it doesn't have to be. So the cyclization occurs when this hydroxyl attacks the carbonyl carbon. This hydroxyl oxygen then becomes part of the ring. So when we see these ring structures here, we see that this now is our hydroxyl oxygen, and our carbonyl oxygen, or our anomeric oxygen, has become a hydroxyl. So there's 2 cyclic forms here, alpha, designated alpha, and beta. Remember, these are both D-glucose, again, based on the structure around that penultimate carbon. But we see that there's an alpha form and a beta form. And the easiest way to think about that, although it might not be physically the most correct way to think about it, is that this hydroxyl attacks from above the plane of the forming ring, pushing the anomeric carbonyl oxygen down out of the plane of the ring, so that's the alpha form. In the beta form, you can think of that hydroxyl as attacking from below the structure of the ring, and thereby pushing the carbonyl oxygen up above the plane of the ring. If that doesn't work for you, you can simply remember that alpha anomers have the hydroxyl below the plane of the ring in the Hayworth projection, and the beta have it above the plane of the ring. We see that in water, the beta glucose is the favored organization, and that has everything to do with steric hindrance. When we see that in this Haworth projection, this is not a really physically relevant structure. It's just simple to draw because we know that the ring is not planar, and these carbons are sp3 hybridized and tetrahedral. So what looks like a hydroxyl projecting above the plane of the ring in this beta form is actually at the equatorial position. So we see that we have axial positions here, here, and here, and otherwise we have equatorial positions here, here, and here, and out here. So the bottom line is that in this cartoon I've drawn for you here, we see that all of the hydroxyls project out into the equatorial positions. That should give you a sense of why steric hindrance is determining the preference at equilibrium for glucose to adopt the beta configuration. If those hydroxyls, those big bulky hydroxyls, were placed at axial positions, they would be crowding one another above or below the plane of the ring. So we avoid that by populating preferentially this other state. The extent to which the steric hindrance is important is determined again by the relative proportions of 36% versus 64% for the alpha and beta, respectively. You'll also note another piece of nomenclature here, that is, we've got the D, yes, we've got the alpha, yes. We also see pyranose. What is that all about? So this is alpha, right? Again, referring to the configuration of that anomeric carbon, the alpha D glucopyranose, so it's glucose. The pyranose derives from what the overall structure looks like. This looks like a pyran structure, and therefore we'll call it a pyranose. On the other hand, if we formed ribose is perfectly capable of forming ribofuranose, so now we see that instead of a 6-membered ring, we have a 5-membered ring, analogous to the furan structure, so this would be ribofuranose. So Haworth projections, anomeric states, pyranoses and furanoses, and in terms of those anomeric states, steric hindrance determining our preferences. And there's an equilibrium process here in which we can move between states. That's called mutarotation. As your book makes clear, if you began with a solution of all beta D glucose, it would rapidly equilibrate to give you these same proportions again via mutarotation, which is just the opening of the ring and the repopulation into the preferred set of configurations. A little bit more on monosaccharide conformations. I already mentioned that steric hindrance was important, and I think it's really clear here, especially when we compare the beta glucopyranose structure with this chair structure with this boat structure. We see that again these hydroxyls are all projecting out into equatorial positions, and in this case we might be a little bit more crowded based on the placement of these hydroxyls at equatorial positions. So, but this is strictly speaking, that's an allowed configuration, but steric hindrance is going to determine what we form. In terms of the furanose structures, here we see the 5-membered ring structures. There's also deviation from planarity in these rings. Obviously, again, tetrahedral carbons are not going to form a planar structure with a 5-membered ring. Instead, we call these endo or envelope or puckered configurations, and you can see that we can either pucker around the C2 position or the C3 position. And here, C3 position is puckered out of the plane of the ring here. C2, I might as well go backwards and say that, remembering that if this is the hydroxyl of the penultimate carbon, right, there's the penultimate carbon, its hydroxyl has attacked the carbonyl anomeric carbon of the sugar, remember, then this is going to be a beta configuration. Likewise, this is a beta configuration, but this is the anomeric carbon, and it also happens to be carbon 1 in this. This would be an aldose in that case. So we see that carbon 1, and then carbon 2, carbon 3, carbon 4, penultimate carbon, carbon 5. So C2 is popped out of the plane of the ring here, and here we see carbon 1, 2, 3 popped out of the plane of the ring here. So I'll just point out the fact that we can populate either of those structures, and the extent to which we populate one or the other depends on steric hindrance. Moving on quickly to disaccharides, the key notion here is that we link these carbohydrate monomers together via an O-glycosidic bond. This is sucrose, which is alpha D glucopyranose attached to beta D fructofuranose. and pointing out to you the fact that fructose is a 1, 2, 3, 4, 5, 6, right? Also a hexose, but forming a 5-membered ring, whereas glucose, the hexose, is forming a 6-membered ring. I want to point out as well some details, of course, that this fructose is a what? Aldose or ketose? Well, the anomeric carbon is at position 2, so it is a, it is here at position 2, it is a ketose, whereas glucose is an aldose. I'd like you to draw those structures for yourself and think that through. But the other important thing that I want to point out here is that this parenthetical notation tells us where the glycosidic linkage occurs. So the glycosidic linkage involves a carbon and an oxygen that was a hydroxyl and another carbon. Note that this is not a physically relevant structure, this sort of box structure here. Instead, there's a single bond between the carbon here, this carbon and this oxygen, and between this carbon and this oxygen. And the numbers denote the fact that carbon 1 of the first carbohydrate is engaged through a glycosidic bond with carbon 2 of the second carbohydrate. Key here is to realize that the sucrose molecule is not drawn in a standard Hayworth projection, which would place the anomeric carbon on the right side. Please note that the anomeric carbon is on the left side for the fructofuranose. And if you were drawing this a little more carefully, you would see how difficult it is to draw this molecule in a physically accurate configuration. So instead we make a lot of shortcuts in carbohydrate structure sketching so that we can flip these guys around to draw for conveniently drawing the glycosidic bond. So again, this is carbon 2, the anomeric carbon of the fructose. This is the anomeric carbon of the aldose. So we have a beta, alpha 1 to 2 beta configuration. In this case, it's also important to note that both anomeric carbons are engaged in the glycosidic bond. What this means is that this molecule won't ring open, right? The glycosidic bond is effectively holding it in its closed configuration. As compared to lactose, in which we have the first monosaccharide is a galactose. Again, we're not going into the details of all these structures in this lecture. You can look those up if you're interested. So a galactose linked to a glucose. So here's a galactose moiety and a glucose moiety linked through a beta, right? This is beta 1 to 4 alpha. So the alpha, here's the alpha. In this case, the anomeric carbons are both in their sort of standard positions on the right side of the carbohydrate. So the key is that in this glycosidic linkage, a beta 1 to 4 linkage, again, not a physiologically relevant bond structure here, this sort of zigzag or Greek key looking structure, but instead the key here is that we link to a non-anomeric carbon of the second carbohydrate monomer. So in this case, this molecule might still ring open, whereas this molecule the anomeric carbon is engaged in the glycosidic bond and it's closed. So what this means, though, of course, is that this molecule might engage in another glycosidic bond elsewhere, and we'll encounter that in just a moment. Right. We'll encounter it right now, in fact. So let's look at some larger structures of carbohydrate polymers. Fuel storage polymers in mammals are usually referred to as glycogen. We'll see some other ones in a few moments, but here's glycogen. It is a branched polymer. We won't go into the degree of branching or the size of these polymers. Again, that's in your text, but let's just point out the fact that here is in this case, a glucose alpha 1 to 4 linkage, glucose alpha 1 to 4 linkage, and the branch point occurs through an alpha 1 to 6 linkage. So both carbons 4 and 6 are engaged in the glycosidic bonds, and here we get our linear chain, and then branching off from our linear chain we can form an alpha 1 to 6 linkage. It's really interesting to note here that even though the beta anomer of glucose is the one that's preferred, and when we're talking about glucose monomers, when we're talking about this fuel storage polysaccharide, we're talking about the alpha anomer. So get your brain around that one. In any case, this alpha 1 to 4 linkage means that the anomeric carbon of this sugar is ready to form a glycosidic linkage with yet another one. So we see how the polymer can be created here, and we see how the branching can be created through the formation of a glycosidic bond with another hydroxyl. This shouldn't say disaccharides. We're still talking about polymers here. In plants, we refer to the glucose polymers as starches, and we can either be unbranched in the form of amylose or branched in the form of amylopectin. Effectively, these are about the same as glycogen. Amylopectin in particular has a alpha 1 to 4, alpha 1 to 6 branching glycosidic set of linkages. The degree of branching and the size of the polymer varies between animals and plants, but the overall organization is very much similar. We see that amylose, in this particular case, the amylose structure, the alpha 1 to 4 structure, supports the configuration of the polymer into an overall helical configuration, which is interesting and very different from cellulose, which is an unbranched polymer of glucose involved in beta 1 to 4 linkages. So still glucose, still 1 to 4 linkages, but now the beta configuration, not the alpha. What does this mean? Well, cellulose is an important structural polymer because of its linear nature and its rigidity. That rigidity arises from the ability of this configuration to engage in hydrogen bonding between monomers here, between the ring oxygen and one of the hydroxyls of the neighboring chain. And we see then that these hydrogen bonds will stabilize a linear form of that polymer as opposed to the helical form of amylose. So we see then a structural basis for the rigidity of cellulose. I'll point out to you again that where we have to be cautious when we look at the structures of these molecules. Here we see this molecule drawn in a more or less standard configuration. Anomeric carbon is here on the right, beta 1 to 4, but this carbohydrate is drawn flipped, so that now carbon 1 is here, 2, 3, 4, right? So again, the anomeric carbon is here, but the molecule has been flipped with respect to this one. So carbon 1 to carbon 4. In any case, there's cellulose. Chitin, same molecule, beta 1 to 4 homopolymer. However, instead of using glucose, we have an N-acetyl modification at carbon 2, changing the structural characteristics of the molecule. So that's all we're going to talk about in terms of carbohydrate structures. The rest of this lecture, I will focus on the carbohydrate biology. And again, we're giving this short shrift, and I understand that, and I'm sorry for that, but you at least will have a brief introduction. The key is that not only do carbohydrates play a role in fuel storage, that is to say the storage or mobilization of glucose, not only do they play a role in creating rigid structures, for example, in cellulose or chitin, but they also play important roles in cell-cell recognition. And this comes from the fact that, first of all, cell surfaces are extensively glycosylated and individual proteins are extensively glycosylated. What is glycosylation? Well, we'll just use that term to describe any carbohydrate modification of a protein or a cell surface. So it doesn't have to be a protein that's carbohydrate modified. Lipids are also frequently modified at their head groups with carbohydrate oligomers. This glycosylation mediates a whole slew of interactions. Intracellular targeting, that is to say, where we localize our protein within the cell, cell recognition, tissue organization, and tissue organization is controlled by cell recognition, invasion of pathogens, or the recognition of an invasion of pathogens. All of these processes mediated by decorations of cell surfaces and proteins with carbohydrates. How do we do that? Well, we have our usual suspect of side chains that get modified through glycosidic linkages to the carbohydrate monomers or oligomers or polymers. Here we see a serine modified through an O-linked glycosidic bond with an N-acetyl galactose, and maybe there's more going on up here, we don't care. And then in this case we can also form an N-linked glycosidic bond with an asparagine. So usually the O linkages occur through serine or threonine. Usually the N linkages occur through the formation of an amide with asparagine. Furthermore, we can make some statements about what this glycosylation, what kind of role this glycosylation plays in trafficking of proteins within the cell. First of all, we can note that N glycosylation occurs in one set of compartments, both the endoplasmic reticulum and the Golgi apparatus, whereas O-glycosylation occurs strictly in the Golgi apparatus. Without going into too much detail, we'll simply say that a specific kind of oligosaccharide tag, right, a specific kind of sort of oligosaccharide tag directs us to a particular localization. In this case, we're just showing that that could mean that we'll go to a lysosome, or to a secretory granule, or to a vesicle that's going to merge with the cell surface. So we see that we can control all of this stuff by the kind of oligosaccharide tag that we've placed on the surface of our protein. We'll come back to that in just a moment, but let's also point out that lipids, which we haven't discussed yet, can also be modified at their head groups by oligosaccharides, and we see that the identity of the oligosaccharide can determine things like blood type. So there's a lipid down here that we're not worrying about right now. Instead, we'll forget about that and just assume that the lipid is part of the membrane, the outer leaflet of the membrane, and what the membrane presents now is this carbohydrate, which we've color-coded as galactose or N-acetylglucose or fucose as these colors. And we see that the blood type, as determined by whether or not your own antibodies recognize one structure on your cell surfaces or not, is simply determined by the structure of this oligosaccharide. And we see that there's a core structure that's the same between O, A, and B, right? It's this galactose, N-acetylglucose, galactose fucose, but we see that there's a branch point that can happen at that third position that can either be an N-acetylgalactose or a second galactose, and that difference determines, in part, your blood type. Okay. Back to proteins, however. How is this recognition carried out and mediated by carbohydrates? So let's just simply say that there are a class of proteins known as lectins that recognize cell surface carbohydrates. So we can assume then that the organization of carbohydrates on the cell surface can be very specific, and that there are specific classes of molecules dedicated to looking for and finding the proper set of interactions between cells. So the carbohydrates then on the cell surface and the lectins that recognize them mediate interactions between circulating proteins and the receptor molecules. They also recognize the proteins that can be marked for degradation. So an important idea that we're not going to spend nearly enough time with is that protein glycosylation can affect a protein's half-life and can serve as a marker for degradation. A class of lectins called the selectins are displayed on leukocytes, that is to say circulating white blood cells, and they actually can mediate motion along the endothelium of the blood vessels. I want to remind you of one of the first lectures that we saw in this class, in which I showed you the video from Harvard that showed motion of a leukocyte to the point of an infection. I'm not going to show it again, but I'm going to remind you and ask that you go back and check it out again, reminding you that the localization of these leukocytes to the source of a wound are mediated by carbohydrate recognition motifs. Here just happens to be one called sialoadhesin. It recognizes a sialic acid, which is N-acetyl neuriminate, which is just this carbohydrate here. The structure of it doesn't matter in great detail to us. Just recognize that it's modified, right? We have a carboxylic acid group here. We have another N-acetyl group here. So you get the sense through this structure that the carbohydrate structures can be very, very complicated. In any case, here's sialoadhesin, and we see looks like a trisaccharide bound at its carbohydrate binding motif, and we see that there's some important tryptophan interactions between the carbohydrate and the protein. I'm just showing you this cartoon to give you a sense of what those interactions might look like. We'll see a couple more in the next couple of slides. Yes, mannose binding protein. This is a C-lectin, also known as a calcium-dependent lectin, presented on the surface of cells, recognizing particular carbohydrates, and in this case we see that the protein is actually a trimer and therefore might recognize a multivalent binding site. And this is a way not only to enhance the affinity for targets, but also to enhance the specificity. So you could imagine, although we're not in this case, you could imagine that the kind of carbohydrate recognition motifs you present on your trimer could determine what kind of carbohydrate motif you would recognize on the target. Let's see. And this is a kind of a hard to parse cartoon, but in black here we see some of the carbohydrate binding determinants of mannose binding protein, and there's the calcium ion, and just showing in detail here. Even though we're binding monomer carbohydrates, here we see something that's more standard, which would be perhaps the antigen that's presented on the surface of an infected cell, or presented on the surface of the infecting cell, perhaps either or both of these. And we see that, the only point in showing you this is that, okay, we have an N-acetyl glucose, we have a galactose, we have this other crazy carbohydrate here, and we see that these interactions are mediated by the usual suspects. It's all I'm trying to get at here. We have this calcium that's interacting with some hydroxyls on this second carbohydrate. We see this lysine interacting again through some standard interactions, some of which are mediated by a water molecule with the carbohydrate itself. So nothing new there in terms of what stabilizes these interactions. In terms of some other biology, we can look at sialic acid again and think about how it mediates the adhesion of viruses to the cell surface and the entry of the virus into the cell. And the particular target we're looking at here, and you can go read about this on the PDB Molecule of the Month page, is hemagglutinin, which is effectively a lectin that's presented on the surface of influenza virus that mediates adhesion to the target to a target molecule, to a target cell. And there's this process is broken up into a few phases, and I think this illustrates a lot of the concepts that we've discussed over the course of the term in addition to carbohydrate recognition. So effectively, the hemagglutinin presents in this blue structure a structure that recognizes the carbohydrate presented by the cell membrane. So there's your sialic acid recognition event. But what happens upon binding is the delivery into the endosome of the hemagglutinin molecule, and therefore the virus. Remember, we're attached to a virus down through hemagglutinin. So we bring the molecule into the endosome, but that acidic environment induces a conformational change that exposes these extremely hydrophobic portions of the chain, which prefer to be buried in the membrane. So we have multiple things going on. Acidity reshaping the structure, the reshaped structure involving exposure of hydrophobics that insert into membranes, facilitating membrane fusion. So now remember what's happened is that the virus has been brought in wholesale into the endosome, but now because of the organization of these hydrophobic elements, membrane fusion can occur, and the contents of the virus itself can be spilled into the cell. That's infection. So again, in this case, we're not talking about how the immune system recognizes infected regions or infecting agents. We're talking about how the infectious agents themselves utilize carbohydrate structure on the surface of their target cells to gain entry. I have a couple more structures to show you about viral entry. And these are articles that you can go read if you have time or interest. I'm really showing you these because, again, these are the sort of events that we're used to seeing, and so the point being that the carbohydrate binding is no different in kind than any of the other intermolecular interactions that we've discussed over the course of the term. So let's ground ourselves here. In yellow, we're looking at a carbohydrate that is bound to a receptor molecule. The receptor is presented on the surface of the cell. What is it a receptor for? It is a receptor for hemagglutinin, as presented by the human virus. This happens to be the hemagglutinin based on the sequence of the influenza virus from the 1934 disease, and in green, so in green we see the hemagglutinin molecule, and we see the extent to which it forms interactions between with the carbohydrate itself. And again, those are partially mediated through sialic acid, but there are other carbohydrate interactions as well. And so that's the main thing to show you here. I guess the point in this article was to try to think about what changes in hemagglutinin have taken place from year to year, and what changes in particular occur in particularly bad influenza years, for example, when avian viruses can hop over to human hosts. And without going into those details, suffice it to say that it has everything to do with how that hemagglutinin recognizes the receptors on the carbohydrates on their host receptor molecules. One more thing to say about carbohydrates before we leave them. Still focusing on antivirals, we've talked a lot about inhibitor molecules and how inhibitors affect the ability of enzymes to do their job. This is sialic acid, right? It's a carbohydrate, heavily derivatized again, but here are some antivirals, and you can see again why these might be good inhibitors just based on the structural similarities they have to the natural target. In this case, these are inhibitors of neuraminidase. This is a carbohydrate binding molecule that's also an enzyme. It actually cleaves the carbohydrate moieties from the proteins which they decorate, and neuraminidase is involved in maturation of the virus. So when you have a newly developed viral particle on the interior of the cell, it's interacting with the interior cell membranes through carbohydrate lectin interactions. In order to release that virus into the environment, those interactions have to be overcome, and they're overcome by neuraminidase cleaving those carbohydrates from the cell surface. So there's a couple of things to show you here. First of all, as I said, sialic acid has some structural resemblances to those drugs, and these drugs are, I should point out, derived directly from crystal structures of neuraminidase bound to sialic acid, or sialic acid derivatives. In terms of understanding how the viruses develop resistance to these drugs, here is a particular, I've highlighted here in red, a particular substitution that occurs in drug-resistant strains of the virus, a histidine to a tyrosine. And it's a little bit difficult to see here, but you can see that the histidine has been mutated to a tyrosine. Tyrosine is in blue, and here the key interactions are determined by this glutamic acid. And let's just point out that, first of all, there's a lot going on in the structure, so let's look at it carefully. Here we see neuraminidase in the green sort of chewing gum structures, but in terms of our ligand molecules, in gray we can sort of barely see sialic acid, the natural molecule. In green we see zanamivir, and in yellow we see oseltamivir. And the key is that this histotyrosine change results in the movement of this glutamic acid, which it looks like a very subtle movement, right? But what occurs is a change in the affinity for oseltamivir, and this substitution results in a virus that's resistant, in particular to this drug. And you can see that it's because, probably, or these authors posit, that there's steric hindrance between the new position of this glutamic acid and, as stabilized by this hydrogen bond with tyrosine, now tyrosine 274, that it's a disruption that doesn't occur in zanamivir because the structures are so drastically different. So the point with this slide being that we have multiple drugs that we've developed based on their similarities to sialic acid and their abilities to inhibit neuraminidase, but that viruses undergo rapid evolution to overcome those drugs, and we can structurally rationalize what those changes are. I think I have So in this slide, we see now, likewise, this is just simply a single drug, oseltamivir, and we see again Now what we're looking at is the same drug bound in either the wild type or, again, this His 274 to tyrosine substitution. And we see that the changes are subtle and have to do with the configuration of this end of the molecule, which is this end of the drug. So this subtle change, histidine to tyrosine, well, perhaps not so subtle change, histidine to tyrosine, induces a change in the configuration of the bound state of this drug, which lowers effectively the affinity of neuraminidase for that drug, thereby reducing its effectiveness as a drug. So I'm going to end our carbohydrate discussion today. We've taken a meteoric rise from definitions of carbohydrate monomer structures to interpretations of carbohydrate-protein interactions. And I know that it was whirlwind, but I hope that it provides a basis for you to think about these interactions in a little bit greater detail.